Good afternoon, everyone. I'm Christian Cordero, and I'm one of the technical assistant at Fish Health Section. And this afternoon, I'll be presenting to you about fish health diagnostic services. And also, this will serve as a reorientation on the different aspects of sending samples, fixed and live samples for disease diagnosis at our laboratory. So to begin with, these are the different diagnostic services that we are conducting or we are performing in the laboratory. We have the bacteriology and mycology, the PCR-based diagnosis, direct microscopy, histopathology, and aflatoxin detection. So for the bacteriology and mycology part, we have bacterial isolation, wherein we can conduct different isolation from biological samples, and also we can do that one with your water and soil sample. For bacterial count, we do have luminous bacterial count, total plate count, and presumptive vibrio count after we do the isolation process. And then right after, we can also conduct bacterial identification. So right after isolation, we can collect some isolates or some bacterial isolates in some of our isolated bacteria. And then we can do series of biochemical tests for that one. And then we can, again, identify your bacterial isolate up to the genus level. And then for the marine fungal isolation, we first observe some cottony-like structures in any parts of the diseased animal, and then right after we wash the tissue in sterile water, and then during the isolation part, we isolate that one using your PYG or your peptone yeast extract and glucose agar with antibiotics, and then we do the identification based on the fungal morphology. That is, we check the structures of your hyphae and some fruiting bodies like your conidia. Then for PCR-based diagnosis, we do conduct conventional PCR and real-time quantitative PCR for your fish and your shrimp and crustacean in general. For shrimp and crustacean viruses and parasites, we do conduct white spot syndrome virus, infectious hypodermal and hematopoietic necrosis virus, your monodon baculovirus, your yellowhead virus, or sometimes we call that one as gill-associated virus, your Taura syndrome virus, whitetail disease, your enterocytozoon hepatopene, acute hepatopancreatic necrosis disease, and your infectious myonecrosis virus. And for fish, we conduct viral nervous necrosis, tilapia lake virus, koi herpes virus, spring viremia for carp virus, and red sea bream iridovirus. That's for your conventional PCR. And for real-time quantitative PCR, we do conduct white spot syndrome virus and infectious hypodermal hematopoietic necrosis virus for both biological sample and water and soil also. So for direct microscopy, we do conduct fish and shrimp larval monitoring, MBV detection, parasite detection, and identification. For fish and shrimp larval monitoring, we do check some of the growth and development factors or changes in your shrimp and fish larvae. At the same time, your physical appearances and the different types of physical aspects in your fish and shrimp larvae. We are also conducting monodon baculovirus detection via direct microscopy. So we will try to dissect the hepatopancreas of the shrimp, and then right after we can fix that one with malachite green, and we'll do the direct microscopy to check for the different inclusion bodies present in the sample. So by the way, these inclusion bodies is part of the infection of your monodon baculovirus in the shrimp. Then for parasite detection and identification, we can dissect the fish or isolate some mucus sample in the fish and then check for the parasite. And then right after, we can check the identification via the different attachment organ or the different morphologies of your parasite. Then for histopathology, we do conduct typical section and serosection. For typical section, this is the regular type of, or the most requested type of sectioning in the microtechnique laboratory, in which you can select specific sample from the whole block of tissue that you have prepared or that you have fixed. And then for the serosection, aside from the whole block, you can also have vast amount of samples, since you can directly acquire whole sample or the whole cut in your whole tissue being fixed in this sectioning. Then for aflatoxin detection, we do have the aflatoxin rapid test kit from eLab Science. So this kit uses the principle of colloidal gold immunochromatography assay. This is usually an antigen antibody detection or interaction that is specific for your aflatoxin. And then the detection of your total aflatoxin in samples will be from your grains, your feed, and some of the muscle tissues in your fish or in your crustacean. Then let's move on to the methods of sending samples in the laboratory or in our laboratory. These are basically the different types of samples that we are receiving in the laboratory. So basically we have, for example, for crustaceans, we do have shrimp and crab. For fish, we have grouper, milkfish, snapper, and pompano. For mollusks, we sometimes have oyster and mussel. And then for the regular type of monitoring that we are receiving in the laboratory, we do have the water and the soil. So for shrimp, when you try to send live samples in our laboratory, you need to obtain 3 to 5 deceased or moribund shrimps and an equal number of normal shrimps. So basically, this is important so that we can compare the normal ones from the deceased ones. And then you need to pack it separately and aerate or place oxygen in the bags. And then place the bag in a styrofoam box to keep temperature cool. And at some point, you can place some small packs of ice when traveling long distances or during hot weather. Or if shorter distances, you can have an insulated that one in a styro box with oxygen or even without the ice. Then for fixed samples, this one is for the PCR analysis. Samples should be fixed in 95% ethanol. It should be placed in a tightly capped bottle or plastic container to avoid leaks. And then for large specimens, you can dissect directly your sample and you can dissect the gills and you can fix that one in 95% ethanol. And please do remember that the sample should be submitted to the laboratory as soon as possible to avoid the degradation of your sample. So for fixed samples for histopathology, you can use Davidson Fixative at 0.1 to 10 ml depending on the size of the shrimp. So the bigger the size of your sample, you need also bigger amount of liquid or fixative that you'll be using for your sample. So you'll be using syringe to inject the different, the fixative into the different areas or the target areas of your shrimp, for example. So you can always do this procedure if you will have the idea on how to inject the fixative on the different surface, the different parts of your shrimp. And also if you have access to the different fixatives in your laboratory or in your hatchery, you can always do this procedure. The main idea of why we need to inject the fixative into your shrimp is to basically fixate first the inner parts of the target tissue. For example, if we have hepatopancreas, need to inject directly the fixative so that the inner parts of the hepatopancreas will be fixed with the fixative. Since it's very hard for the fixative to penetrate within the inner parts of the tissues. The more the case that we have, for example, if we have larger fish or larger tissues, it's really hard for the fixative to penetrate within the inner parts of the sample. As I said a while ago, for larger samples, you need to carefully fixate your sample and then at least inject some fixative within the bigger sample since the penetration of the fixative is very important. And then you can cut it and make some incisions that could be carefully, that you could carefully collect your sample upon fixation. Then let's move on for fish. So for fish, we do accept live samples, ice samples, or sometimes fixed samples. So for live samples, we do conduct microscopy, bacteriology, mycology, and your molecular part, which is the viral detection. And then if you fail to send some live samples in the laboratory, you can always use iced samples. You can also conduct microscopy for that one, bacteriology, mycology, and virology. And then for fixed samples, we do, again, PCR and then histopathology. So if you will try to send live samples, again, we need to at least secure 3 to 5 week fish or deceased sample, and then again obtain 3 to 5 normal fish or healthy fish, and then you need to pack it separately, the normal ones from the deceased ones, and then you need to transport this one in a water-filled plastic bags with sufficient aeration. And then the water volume should be at least 20 parts of water into 1 part of fish. And then you should always use double bags if you have larger samples to avoid leakage of the sample. And then place bags in an insulated or styro box when transporting. And then again, for longer distances, you can use ice. If shorter distances, you need to at least secure your plastic bags with aeration. Ample amount of aeration would do. And then do not add too much ice so that it would not make the water very cold, or the temperature in your packed sample would not be very cold. And then always consider this one. Make sure that the specimens arrive to the laboratory live. Then for ice samples, if you fail to submit live samples, you can use always ice samples. You need to at least obtain 3 to 5 deceased and normal fish again, and then you need to wrap them again individually, and then separate your normal from your deceased ones. And then again, place your pack samples, this time between layers. You can use plastics wherein you place your fish, and then you need to layer that one in the styro box, so that when sending the samples, you have a very thorough iced sample during transportation. And then again, you need to consider that ice samples should arrive to the laboratory for analysis within 24 hours. Then for fixed samples for your fish, you can always use 10% buffered formalin. If you have an access to that one in your laboratory or in your hatchery, you can always do the preparation of your 10% buffered formalin, and then you can use that one for fixation of your sample. You just need to obtain 3 to 5 deceased and 3 to 5 normal fish again. And then since your fish is very agile, you need to at least kill them or immobilize them. You can use MS-222 and 600 ppm of your phenoxyethanol so that you can immobilize or kill your fish. And then you need to open the abdomen. So this one is upon dissection, the large fish for its entire length. And you need to immerse, again, if you try to collect the tissue, you need to immerse carefully your specimen or your tissue sample in the fixative, in which you have a ratio of 1 part fish tissue to 10 parts of your fixative. So that's, for example, you have 100-gram fish, you need 1,000 ml of your formalin solution. And then for larger fish, you can always cut different types of tissues into several parts so that it's very convenient for you. For this one, if you have a smaller bottles or bottle container, during fixation, you can always cut the different tissues into smaller ones. Then make sure containers are properly sealed before sending to the laboratory or to our laboratory. This one is to prevent leakage. So for fixed samples, for the molecular level of detection, that's for your viral detection, you can always disinfect your tray and then sterilize your paraphernalia upon dissection. This one is to avoid contamination during the dissection process. And then upon dissection, you need to at least secure organs like your brain and then your eyes, for example, for your viral nervous necrosis detection. And then you need to carefully collect those tissues for us to have a whole sample. And then you need to fix that one in 95% or 70% ethanol. Then upon sending your fixed samples into our laboratory, you need to include some of the informations that we need. And then like, for example, your signs of disease that has been observed in your hatchery in your area, the patterns of mortality, the date of disease signs initially observed, the water source, the water management, and some of the quality control that you're using in your laboratory or in your area, your feed and feeding behavior, the species, the size, the age group affected, also the sketch of your farm layout, and then the number of ponds or cages affected in the whole farm, the new innovations in your farm, and some of the farm or industrial activities in your neighboring area. Then for your water sample should be taken as soon as the fish begin to act abnormally and before the water change was done in your area or in the hatchery. So you need to send at least 100 ml of that water, and then you can use sterile or boiled bottle container. So if you don't have any types of sterilizing material like your autoclave and pressure cooker, you can always boil your water and then disinfect that one or sterilize that one in your container. And then right after, you can put the right amount of water sample you'll be needing for the diagnosis. And then always remember that the sample should reach the laboratory within 2 hours of your sampling. These are some of the important notes that you need to remember upon sending your samples into the laboratory. So you need all pertinent or data information must be sent to the laboratory, and you need to at least notify the laboratory of the delivery and arrival of the samples. And then your fish or your biological sample should be sent to the diagnostic laboratory by any of the method mentioned, but we usually prefer it live. And then it is best to send samples live for our parasite detection, for the bacterial isolation, for the viral detection, fungal isolation, and then histopathological studies that can be done in your live samples. And then when sampling fishes from different tanks and ponds, be sure to use separate clean gears like fish nets and pails, etc., to prevent cross-contamination between tanks and ponds. This one is very effective when you have already determined the ones that are diseased and the ones that are normal. So if you have not yet determined the diseased ones from the normal ones, you can always use different paraphernalias each tank. Like each tank, you need to use fish nets and then pails separately from the other tanks. Then we do have this different types of diagnostic services fees. You can always check this diagnostic services fees at SEAFDEC.org.ph, wherein you can have your, or you can update yourself of the different types of fees that we are trying to collect after you request some of the diagnoses that you're be needing for your sample. There we have the different types of prices for your direct microscopy, for your PCR-based diagnosis, and then for your bacteriology and mycology, and then for your histopathology and for aflatoxin detection. And then these are also the different types of forms that we are trying to fill upon the request of the one who sent the sample in the laboratory. So first, we do have the official diagnostic form, wherein you can use that one as an initial paper or initial form, wherein you need to try to fill up the time of the sampling, the different types of analysis that you will be requesting in the laboratory, and then also some of the different signs and symptoms that you have observed in your sample. And also for your request for your fish, we do have this form in which we can check the different types of morphological changes in your fish, especially those the different tissues in your fish upon dissection. And then we'll try to consolidate that one later, and then we'll have a final diagnosis for your fish. And then also for your crabs, we do have also almost the same type of diagnosis form, but since we have different types of tissue samples in your shrimp and in your crabs, we do also check those types of changes upon sending your samples in the laboratory. And then also for the fish and shrimp larval monitoring, we do have this form wherein we try to check the different types of growth and development of your shrimp and your fish larvae upon sending your samples in the laboratory. And again, you can always visit seafdec.org.ph, or you can email us at fishhealth@seafdec.org.ph, or you can also try to contact us at these numbers, wherein you can also check some of the details of sending your samples in our laboratory, or if you have any comments or suggestions to improve our diagnostic services in the laboratory. And thank you for listening.